I haven't really mentioned what miRNAs are. Again, I try to make this a balance between a more advanced talk, but a more introductory talk for those who may want to get into the field of microRNAs. So I've kind of already mentioned this, what miRNAs are. They are regulators of survival and proliferation pathways. They're very important in normal development, and they essentially dampen a lot of cell signaling networks, and they do this in a cell context-dependent manner because essentially they're operating in feedback loops. They're regulating transcription factors, and those transcription factors are obviously regulating a host of other genes. But those transcription factors can feed back and regulate microRNAs. So they're kind of in this unique little network, if you will. So where they come from, microRNAs themselves are transcribed from individual gene loci. They can have their own promoters, so they can be intergenic, or they can be intragenic, so they can be in an intron of a host gene. When they're transcribed, they make a primary microRNA transcript, and that primary microRNA transcript can be just have one cistron, so it just has one microRNA hairpin, or it can be polycistronic, so something like a miR17 cluster. There's many microRNAs that come off of one primary microRNA transcript. So that's what we mean by polycistronic. But essentially, the way microRNAs work is that they are processed by RNase 3 enzymes that are present in our own cells. So DROSHA recognizes, and PASHA basically recognizes hairpin structure, cleave it, form a pre-microRNA that gets exported into the cytoplasm, and then essentially another family of RNase 3 enzyme, at least in this case dicer, recognizes this loop here, cleaves that, and then you end up with a duplexed RNA. There's selective loading of this duplex into the RISC complex. So one strand is selected, and it has to do with the instability, I guess, of the 5' end of the microRNA duplex. If this, I've tried to show it here, where if this strand has a free 5' end, this gets loaded into the RISC complex, this one strand. And so this essentially what this microRNA does is it serves as a guide, and it recruits RISC to target mRNAs, predominantly in the 3' UTR region of a coding gene, but could also bind in other regions as well. And what it does is is recruiting RISC at this RNA-induced silencing complex, which comprises AGO, and some say that you have to have, if you have perfect complementarity, you have slicing activity. If there's a bulge, then in this sort of Watson and Crick base pairing, then you just have stalling and translational repression. It could also be that if different different Argonaut proteins are involved in RISC, then you could maybe get differential effects of microRNA activity. I think that's still a little not clear. But the other point I wanted to make is that essentially, as of right now, at least what's reported in miRBase, it hasn't been updated, I think there's still about 1,800 microRNA precursor genes, and again, some of those are polycistronic, so there's about 2,500 mature microRNA sequences, and really, over the past decade, we've probably been studying just 100 or so, 200, really well. So there's a plethora of microRNAs out there that could be important in anybody's system.
To view the full video of this and all of our other webinars for bioscientists at the bench, please visit bitesizebio.com/webinars.